Hi, hello, good morning. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MVS Government Arts and Science College, Autonomous, Mahbubnagar, Telangana State. In our last class, we studied about photosynthesis, the process where organic substances are produced by the green parts of the plants from inorganic substances. Okay, so these substances which are formed as a result of photosynthesis are to be translocated to different parts for their usage. So this process is known as translocation. We all know that the plants, the flowering plants, and the gymnosperms, and these angiosperms, gymnosperms, and pteridophytes have the vascular tissue. What do you have? What do we have in vascular tissues? We have xylem and phloem. What is the function of the xylem? The mineral salts are transported unidirectionally from root to the shoot, along with the transpirational stream. So it is a complex tissue, and phloem is also a complex tissue. We all studied in our lower classes the structure of the phloem. Today, in the later part of our class, we will see the structure of the phloem once again. Okay, so um, these organic substances, whatever are formed in the leaves, that is the source, okay, are transported uh, through phloem to the different parts of the plant, that is roots, branches, flowers, non-green parts which do not synthesize food receive this uh, food material through phloem. Okay, this process is known as translocation. Long distance transport between organs through vascular tissues is called translocation. Or more commonly, the transport of organic solutes through phloem is known as translocation. So we will see how this translocation occurs in the plants. Okay, so evidences in support of phloem. We said that through phloem, these organic solutes translocate from the source to the sink. So, what are the evidences? How can you say that the phloem helps in transportation of these organic solutes? So, experimental evidences in support of translocation of sugars through phloem are established. Okay, the first one is a ringing or girdling experiment, analysis of sap from aphid stylet, evidences from tracer techniques, and evidences from chemical analysis. All these evidences support that the translocation of sugars takes place through uh, phloem. The first one is ringing experiments or girdling experiments. We also call it as girdling or ringing experiment. The girdling or ringing experiment is a method of breaking the continuity of the vascular tissue. You can see here clearly, see the cursor, the bark and the vascular tissue has been removed. The external vascular tissue has been removed from the stem. Okay, so here uh, what it means, it means it breaks the continuity of vascular tissue by their removal. Okay, so in girdling technique, a ring of bark is completely removed from stem or branch with wood left intact. The ringing of phloem breaks the downward movement of organic solutes. Downward movement. We all know that the xylem transports the water and minerals from roots to the shoot system. In the same way, the phloem, the transportation through xylem is unidirectional, but the transportation through phloem is bidirectional. We will learn it again. So for now, uh, in these ringing experiments, the ringing of a phloem breaks the downward movement of the organic solutes. So what happens? All the organic solutes which are formed at the source, that is at the canopy or green parts of the plant, they transport to different parts. In the same way, they transport towards the roots, towards the stem. So here the phloem is removed. So what happens? No way, no path for the solutes to travel downwards. So they accumulate here at the top of the girdle or ring that we made. Okay, so you can clearly see that here there is a clear bulging of the stem. That means accumulation of organic solutes, and slowly the lower part it will get dry and it will die. Okay, so this shows that by the removal of the phloem we can stop the translocation of organic solutes. Okay. In the same way, the next experiment, that is analysis of sap from aphid stylets. Okay, aphids are the insects. They draw sugary phloem sap. That is, sugars. All the sugars are translocated through phloem. So the phloem is rich with the sugary substances. So these sugary substances are drawn by these aphids. They use their stylet. They pierce their stylet into the sieve tubes of the phloem, and they draw the sugary substances. Okay, so they puncture the sieve tube, um, and during this experiment what they did is that the feeding aphid is anesthetized with carbon dioxide. They supplied the carbon dioxide to this feeding aphid, and it slowly anesthetized, and they separated this aphid from its proboscis. What is proboscis? Proboscis is the small, narrow, tube-like structure which is used as a straw. Okay, it pierces that proboscis into the sieve elements of the phloem, and it draws the sugary substances. So by anesthetizing it with the help of carbon dioxide, these aphids are separated from the proboscis. You can clearly see here, this is the proboscis. Okay, it is pierced into the uh, sieve elements by puncturing the stem. Okay, so now as there is a hydrostatic pressure in the sieve tubes, the exudate comes out of the cut surface. Exudate means the whatever sugary substances that are present inside the sieve elements, they have some hydrostatic pressure. So this proboscis acts as a small outlet to them. So these sugary substances slowly they come out as a uh, cap as a water or whatever solution. If we keep what do you call it, mm, capillary tube, it will flow out. So in the same way here also, the proboscis, through the proboscis the sugary substances oozes out as an exudate. Okay, so these exudates are collected in the micropipettes, and they are analyzed by chromatography techniques. They are analyzed. Okay, so uh, as a result of this chromatographic analysis, you can find that all the sugary substances, that is sucrose elements, are present in high concentrations in the sieve elements, that is in phloem sieve tubes. Okay, this is also one of the evidence in support of the phloem. And the third one is evidences from tracer techniques. What are tracer techniques? Tracing using radioactive elements. If you use radioactive carbon dioxide, then the organic substances whatever formed, they also contain this radioactive carbon. And this radioactive carbon, which first forms as glucose, later on converts into non-reducing sugar, that is sucrose, and it is translocated only in the form of sucrose but not in the form of glucose or fructose. Okay, so this non-reducing sugar which is formed from the glucose, it also contains the radioactive carbon. So if we trace this radioactive carbon, it shows that the phloem is the structure which helps for translocation. That means there in phloem you find the uh, sugar substances in higher concentrations than in xylem. Okay. Then the fourth one is evidences from chemical analysis. Chemical analysis of phloem and xylem contents reveal that the phloem tissue has a higher concentration of carbohydrates and organic nitrogen compounds when compared to that of the xylem. So these are the evidences in support of phloem. Okay, that is phloem is the structure, is the complex tissue which helps in translocation of organic solutes. Then direction of translocation. We said that earlier. Xylem is unidirectional. That means water and minerals are translocated from, they are absorbed in the roots and they are translocated through the upper parts, and it is unidirectional, and that is against to the gravity. Okay, but here the phloem, the organic solutes are translocated bidirectionally. That means they they are transported towards the lower parts, as well as they are transported towards the upper parts, such as flowers or fruits, anything which are present at the apical regions of the plant. Okay, so we can summarize that movement in the xylem is strictly unidirectional, that is upward only, and uh, downward movement of these minerals and organic solutes is through phloem. Organic movement, organic compounds move in both direction in phloem, that is they are bidirectional. And the direction of phloem transport is generally towards growing regions. In a young vegetative plant, the metabolites are transported rapidly to the growing root system, shoot apices, and developing leaves. Hmm? As the plant grows in size, lower leaves transport mainly to the roots, whereas the upper leaves transport to the growing shoot apices. Okay. Source and sink relationship. What is source? Source is nothing but where the production occurs, where the storage occurs, and sink where these products are utilized. So the phloem translocation, there is a sort of source and sink relationship, which is not governed by gravitation, as the flow in phloem is, as the translocation in phloem is bidirectional. That means it can move upwards also, so it is not governed by the gravitation. Leaves with their photosynthetic capacity constitute the source, and the products of photosynthesis are called photosynthates. Okay, so um, these metabolites move from source to growing and storing organs such as stems, young leaves, flowers, fruits, tubers, corms, roots. All these constitute the sink where they get utilized. Okay. So this is source and sink relationship. Now we see the structure of phloem. You all know that it is a complex tissue. Okay, it's a complex permanent tissue running almost parallel to the xylem tissue. Okay, phloem is made up of four types of cells. You can observe in phloem: sieve tubes, companion cells, parenchyma, and fibers. Phloem fibers. Okay, so you can see here these are sieve cells. These are companion cells. These are phloem parenchyma. Here also you can see clearly these are sieve cells. This is phloem parenchyma. These are companion cells, and these are phloem fibers. These four types of cells are seen in phloem. Okay, so out of these, Sieve tubes are involved in translocation of organic solutes. Very important sieve tubes. You can see the structure here clearly. These are the sieve tubes. These are the companion cells. Okay, so these sieve tubes, sieve tubes consist of elongated cells, hmm, uh, arranged end to end. Elongated cells arranged end to end. These sieve tube members have transverse or oblique end walls called sieve plates. You can clearly see here. These are the end plates connecting two sieve cells. This is also the end plate seen from the upper vision. Okay, so in transverse section you can see like this the sieve uh, tube, a sieve tube, a sieve element, or sieve cells. Okay, so this is the end plates. Either they may be transverse or they may be oblique. These sieve plates in turn have sieve pores. You can see clearly here. These openings are called the sieve pores. Okay, these small dot-like structures are called sieve pores. And the diameter of these sieve pores may range from 0.1 to 5 mm. And these sieve plates and sieve pores are lined with a callose. Callose is nothing but a glucose polymer. Okay, these are lined with callose, and the deposition of these callose makes the pore diameter very narrow. Okay. In mature sieve tube members, you can't find any nucleus or vacuole. They lack the nucleus and vacuole, and only cytoplasmic organelles like plastids, mitochondria, endoplasmic reticulum are present in the peripheral walls. So you can clearly see here no nucleus is present. Here in companion cell you can see a very clear nucleus, but here you can't see any nucleus. So in mature sieve cells no nucleus and no vacuole are seen. Only plastids, mitochondria, and endoplasmic reticulum may be seen, that too peripherally. Okay, these are cytoplasmic strands. So the lumen is filled with a protein called P protein or phloem protein. Phloem protein or P protein, which occurs as slime bodies. Okay, so whatever protein is present inside this sieve lumen is known as a phloem protein or P protein. Okay, so these proteins are called as slime bodies in the younger cells, and in the older cells they are present in the form of what do you call longitudinal strands or fibrils. In the older cells they are present as longitudinal fibrils. Okay, longitudinal strands are longitudinal fibrils. So this is about the sieve tube or sieve cell. One more time. These are the cells through which actually the translocation of organic solutes takes place. Okay, uh, these are called as sieve cells or sieve tubes, and they are connected end to end. And these sieve cells are separated by sieve plates. You can clearly see the sieve plates here. Either they may be transversely arranged or they may be obliquely arranged. And these sieve plates have sieve pores. Both the sieve plates and sieve pores are um, they are plugged with or they are lined with the callose, which is nothing but a polymer of glucose. Okay, so in mature cells you can't find any nucleus or vacuole. Only cytoplasmic organelles such as plastids. See, you can clearly see here all the drop like structures are nothing but, see the circular structures, these are sieve tube plastids. Okay, so plastids and endoplasmic reticulum and mitochondria may be seen peripherally, the two, and the lumen is filled with a protein called phloem protein or P protein, which is present in young cells as slime bodies, and in mature cells it is seen as strands. Longitudinal strands are fibrils. Okay. So the exact function of this P protein is not clear. It can plug the sieve plate pores upon injury or non-functional. If the sieve cell or a sieve tube it becomes non-functional, then such sieve cells will be plugged by this P protein, and another path will be selected for translocation. Okay, and the cells which are adjacent to these sieve cells are known as companion cells, and the companion cells uh, the function of these companion cells is not known. The exact function of these companion cells is not known. You can see a clear nucleus and dense cytoplasm. See, this is the nucleus and this is the dense cytoplasm in the companion cells. They're always associated with the sieve cells. They appear to absorb sugars and transfer them to the sieve element. That is, they help in phloem loading. Phloem loading. That is, they through these companion cells only the sugar and the sugar, whatever sugar has to be translocated, it enters into the sieve cells. So they help in phloem loading. Okay, so this is about the companion cells. And the third one is phloem parenchyma. They are with thin walls and their function is storage and lateral transport. See, these are phloem parenchyma. They are with thin walls and their function is storage and lateral transmission. Lateral transmission means, see, from the leaves these organic solutes has to enter the sieve cells. So through this parenchyma only they enter, and through these companion cells which are very much adjacent to the sieve cells, they enter into the sieve cells. That is, phloem loading has been done. And the last one is phloem fibers. These are with the thick walled cells and provide support and strength. You can clearly see here, these are the phloem fibers. They are with thick walls and they provide the support and strength to the phloem. So this is the structure of phloem. Okay, it is a complex structure, and inside this phloem you find phloem sap. In most plants, this phloem sap is nothing but sucrose. Okay, sucrose is the only carbohydrate transported in the phloem. If at all any other sugars are present, these sugars may be raffinose, stachyose, or verbacose. If any, if they are present in very much small traces. Okay. Um, and as I already said that only non-reducing sugars are translocated through this phloem. Okay, the reducing sugars such as glucose, fructose, and the phosphate derivatives are not found in the phloem. When we do the chemical analysis, we find only non-reducing sugars such as sucrose, but and small traces of raffinose, stachyose, and verbacose also, but not the reducing sugars such as glucose and fructose. And amino acids, potassium cations, organic acids, vitamins, and hormones, together with the viruses, may be present in the phloem sap. Okay, that means along with the sucrose, you can find amino acids, potassium cations, organic acids, vitamins, hormones, viruses, and some other chemicals, artificial chemicals. If we introduce them also, they are seen in the phloem sap. This is about phloem sap. And now we will see the mechanism of translocation. There are different theories of mechanism of translocation. These organic solutes are translocated in upward as well as downward direction through phloem, as I said earlier. Okay, so this mechanism can be explained by different theories. Protoplasmic streaming theory, proposed by De Vries and supported by Curtis, and activated diffusion hypothesis, proposed by Mason and Maskell. Electroosmosis theory, proposed by Fenson and Spanner, and interfacial flow hypothesis, proposed by Hornert, and Munch mass flow or pressure flow theory, proposed by Munch. Okay, the first one is protoplasmic theory. This theory was proposed by De Vries and later supported by Curtis. Okay, according to this theory, solutes are carried by the combination of diffusion and streaming of cytoplasm. Diffusion and streaming of cytoplasm. There are continuous streaming movements in the cytoplasm, and this helps the organic solutes to move towards upwards or downwards, and through the pores they diffuse out into the next cell. Okay, so within the sieve tube substances, um, either they may be transported towards downward or upward along the cytoplasmic stream. Okay, so this is the theory. This is the uh, thing that is explained by protoplasmic streaming theory. Okay, and the chief merit of this theory is that it explains bi-directional translocation. In whatever direction the streaming may be, the protoplasmic streaming in the same direction, the organic solutes diffuse out to the next adjacent cells. So it explains both bidirectional translocation. And the chief demerit or objection to this theory is that the rate of streaming of protoplasm is much less than the rates at which the solutes move through phloem. Okay, we are saying that the protoplasmic streaming helps the movement of the organic solutes, but here when you see the streaming movements are much slow than the rate of translocation of the organic solutes, so this theory has been discarded. Okay, the next theory is the activated diffusion hypothesis, and this is proposed by Mason and Maskell. This theory suggests that the sugars move down from the source to the sink, that is, from leaf to other parts of the plant body along a concentration gradient by activated diffusion. Activated diffusion is nothing but making use of the energy, making use of energy. That is, it requires energy for accelerating diffusion. This hypothesis has no backing of experimental evidence. So this is also discussed. Activated diffusion hypothesis. The third one is electroosmosis theory, and this theory was proposed by Fenson and Spanner in 1957. Okay. This theory proposes that the movement of ions along an electric potential difference gradient through charged pores of a sieve plate, which carries them uh, the water and the solutes. Okay, that is, this theory explains that the ions along an electric potential difference gradient through charged pores of sieve plate carriers with them the water and solutes. Hmm. We see earlier we saw that. See, you can see here. These are the sieve pores, and these sieve pores are charged. They are negatively charged, and inside here you can find the potassium ions in the sieve lumen. That is, in the cytoplasm you find K plus ions, and here these pores are negatively charged. These positively charged potassium ions, they tightly adhere to the water and solutes present here in the lumen. Okay, the organic solutes which whatever are present here, they are tightly adhered to the K plus ions, and these K plus ions move through these passages, that is these pores. Okay, so in this way the circulation of K plus ions maintains an electric potential difference at the CU plate. Hmm. Uh, these pores are negatively charged, and the positively charged potassium ions sweep through the pores carrying the water and sucrose with them. Okay, so each sieve plate acts as a pumping station, boosting the mass flow of sugars along with the sieve tube. Okay, this is what the electro-osmotic theory says. That is, these are charged areas. See, the sieve pores are negatively charged, and inside the lumen, whatever potassium ions are present, they are positively charged, and these positively charged, these negatively charged, they attract the positive charge, and they uh, these positive charged K plus ions, they adhere tightly the organic solutes, and they move, uh, they pass through this negatively charged sieve pores. Okay, so this sieve plate acts as a pumping station, boosting the mass flow of sugars. Okay, and what are the merits and demerits here? Uh, the demerit is that the potassium ion circulation involves high energy expenditure, and the circulated ions create an electric current that can damage the cell contents. Okay, continuous flow of these K plus ions may create an electric current which may damage the cell structure. So this is the demerit which cannot be addressed by this theory. This is about the electro-osmotic theory. And the fourth one is interfacial flow hypothesis. This was proposed by Van den Honort in 1932. Okay. According to this hypothesis, the solutes move along the interfaces between the cytoplasm and the walls, or between the cytoplasm and the vacuoles of sieve tubes, that is the places between the cytoplasm and the wall. Okay, whatever places, whatever narrow places are there, these spaces help the organic solutes to move. Okay, so it is similar to the rapid spreading of oil at water air interface. If you spill oil in the water, you can observe a thin film over the water and air interface. Okay, it acts in such a way. Uh, a, a thin film is formed between a cytoplasm and the walls. Okay, this film would remain intact with the molecules continuously, which are added at the source and are removed at the sink. Okay, so continuously the molecules are added at the source and they are removed at the sink. Okay, do you understand what I'm saying? According to this theory, a thin film of these solutes has been formed between the walls and the cytoplasm. Wall of the sieve element and cytoplasm, there will be a thin film of these organic solutes. Okay? A continuous thin film, where continuously the uh, organic solute molecules are added at the source and they are continuously removed at the sink. Okay, so the movement of molecules along the interface is much faster than diffusion. Okay, and the main objection to this theory is that sugars, amino acids and other substances are soluble in water, and therefore they could not be present at the interface. Okay, there is water in the cytoplasm, and these sugars, sugar molecules, the amino acids and all other substances which are dissolved, which can be soluble in water, this they dissolve. So where is the chance of existing a thin film of these organic solutes? So this is the main objection, and it is not addressed by this interfacial flow hypothesis. Okay. Then the last one is pressure flow or mass flow theory, which is explained by Munch, proposed by Munch. It is a hypothesis. According to this hypothesis, which is proposed by Munch, the solutes are translocated in the sieve tubes due to a turgor pressure gradient. Turgor pressure gradient, okay, which exists between the source and the sink. And the translocation is passive, that is, it does not require any energy. Okay, so the at the source there will be higher concentration of organic solutes, and at the sink there will be lower concentration. Okay, it is this Munch's hypothesis has better explained by uh, osmometer experiment. Okay, so you can see there two osmometers are connected with a tube. See, let us think that this is osmometer A and this is osmometer B. Both these are connected with a tube. Uh, we call it as C. Okay, these osmometers are permeable only to water. Osmometer A contains higher sugar concentration. Higher sugar concentration means it is the source where sugar molecules are produced. Okay, so it has a higher sugar concentration than B, and both are immersed in water. The water container is connected by a channel. You can see here, this is the water container. See, this is the water container connected with the channel. This is the channel. Okay, this is osmometer A, this is osmometer B. Both are connected by a tube called C, and this is inserted in water water containers, which in turn are connected with the channel here. Okay, so this is the source where sugar molecules are uh, produced, and this is the sink where these organic solutes are utilized. Okay, so we will see. This is a closed system, and as the walls of the osmometers are differentially permeable, uh, only water will enter A and B. Okay, so as here at the source the concentration is high. Why the concentration is high? Because due to photosynthesis the formation of glucose molecules takes place continuously. So these glucose molecules, where they are present, we are calling it as source. So here the concentration of glucose is high. So there exists a water potential gradient. See, outside in the beaker you find only water, but here inside you see the sugar solution, that is glucose is present in dissolved condition in the water at the source. So uh, inside the osmometer A, the free water molecules are very less when compared to the water container outside. So what happens here? The water molecules move from higher water potential to lower water potential. So immediately from the container, the water molecules enter into the uh, osmometer A. We already said that they are differentially permeable and they will allow the water to enter into. So the water enters into the osmometer A. As a result, there develops a turgor pressure. Okay, so turgor pressure develops here, and this turgor pressure flows through this tube to the other side. Okay, so Uh, and this pressure is transferred from A to B through the tube. Okay, so the solution will flow in mass from A to B. You can see here clearly the molecules are moving from A to B. So due to the pressure, turgor pressure that is developed here, these molecules move from this. This acts as a driving force. Okay, so this pressure is transferred from osmometer A to osmometer B through this tube. So in this way, this molecules are transferred uh, in mass to the osmometer B. So, Munch suggested that the living plant contained a system comparable to this physical model, where sugars move by phloem loading into the source and of the sieve tube. The total concentration of dissolved solutes will be greater in the source than in the adjoining xylem and uh, parenchyma cells. Okay, so this is the source where the production of organic solutes takes place. Okay, so it is at higher concentration. Because of this higher concentration, a water potential gradient develops here, and here due to higher concentration you can find lower water potential, and outside higher water potential in the water container. So water flows immediately into the source here, osmometer. So this develops a turgor pressure. This turgor pressure drives these molecules along with it to the um, osmometer B, that is sink. In the same way, the point of production, that is from the leaves, these organic solutes are transferred to the sink through this phloem, which is denoted by a tube here. This tube denotes phloem. This is the point where the organic substances are produced, and this is the point, that is sink, where the organic substances are utilized, and both are connected with a tube-like structure, which is compared to a phloem. Okay, so this is what this Munch hypothesis explains. Here, the difference in turgor pressure between the two ends of the sieve tube is the driving force for the passage of the sugar solution in the sieve tube. Okay. And what are the objections or demerits of this pressure flow hypothesis? According to the pressure flow hypothesis, the sieve tubes acts as passive conducting channel. That is, no usage of any energy takes place here. But phloem tissues show high rates of respiration, and ATP is also present in the phloem exudate. That is, high rates of respiration means where respiration takes place high, where energy is required, there that cells perform a high rate of respiration and produce ATP. Okay, the same thing is seen here. Phloem tissue also shows high rates of respiration, and the ATP are present in the phloem exudate, uh, which clearly shows that these ATP are utilized in translocation processes. And this was demonstrated by application of inhibitors, that is respiration inhibitors such as cyanides and 2,4-dinitrophenol 2,4-D, okay, which inhibits the formation of ATP, and ultimately inhibiting the translocation. So this clearly states that the translocation requires energy. It makes use of ATP. If we use inhibitors such as cyanides and 2,4-D, no production of ATP takes place and no translocation is observed, which clearly says that the translocation requires energy. And the second objection is that the pressure flow can proceed only in one direction at a time, but the translocation of organic solutes in phloem is bidirectional, bidirectional transport in the same sieve tube cannot uh, take place according to this hypothesis. Okay, it is experimentally established that different sieve tubes are involved in the upward and downward directions, which is not explained by this uh, Munn's hypothesis. And the third objection is that the mass flow hypothesis suggests the substances should move with same velocity. Whatever substances are present in the lumen of a sieve cell, they all move with the same velocity. But it is observed that a speed of movement of different compounds varies. Okay, so this is also one of the objections. That is, the water molecules, they are moving very slowly, and the sugar molecule, they move a little bit faster than the water molecules present in the source. Okay, that is in the phloem. So this also is an objection which was not addressed by this Munn's hypothesis. So this is about today's lesson. Okay, we will summarize. Okay, translocation of organic solutes. We have seen the evidences in support of phloem, that is ringing or girdling experiment, analysis of sap from aphids dilate, evidences from tracer techniques, and evidences from chemical analysis. And we saw what are ringing experiments. Okay, ringing experiments, we remove bark as well as a part of vascular tissue containing phloem, which stops the flow of organic solutes, and we can clearly see a clear bulging above the girdle here, above the ring or girdle here, which clearly indicates that the flow of these organic solutes is through phloem. And this is the next one, the aphid uh, test. Okay, so these aphids, they insert their proboscis into the sieve elements, and they suck the uh, sugary substances which are present in it. So we anesthetized this aphids by carbon dioxide, and we slowly separate this aphid from the proboscis, and the exudates whichever come from this proboscis are collected in micropipettes, and they are chemically analyzed. They through chromatography techniques, we can analyze whatever substances which are present in this phloem elements. Okay, so and the third one is the structure of the phloem tissue. It's a complex tissue, permanent complex tissue. It has four cells, that is sieve elements, then the companion cells, the phloem parenchyma, as well as the phloem fibers. Okay, so the mature sieve cells, that doesn't have the nucleus and the vacuoles, but the younger sieve cells, they do have the nucleus as well as the vacuole. So these are the sieve plates and sieve pores. Hmm, uh, they are formed in between the two sieve elements which are connected with each other. Okay, so this is about the structure of the phloem. You can see here sieve cells, and these are companion cells, and this is the sieve plate with sieve pores. Okay, and different theories which explain the translocation mechanism of protoplasmic streaming theory, activated diffusion hypothesis, electroosmosis theory, interfacial flow hypothesis, months mass flow or pressure flow theory. Most of the scientists, they uh, they go with this mass flow or pressure flow hypothesis. Okay, they accepted it. So this is about today's lesson, and thank you.